I was thinking about recording and then didn't. Okay, now I'm recording. Um, if you did not predict the lab values, please put that in by 10 o'clock, so like right after class. The question other was write much of an explanation. So that is the other thing that I wanted to talk about, and maybe this will help as we discuss some of the one of the case studies. Is I want to be sure that when you're writing the explanation that you're focusing on um, not the entire process necessarily, but on the specific question. So we'll talk about case study two in that way to see, and maybe that'll help um, Gina, what you think about what you put so far. Because what I noticed with the case studies that I have graded, one and two especially, is that there's definitely a sweet spot in how much and how little information you present in both the exam and the case studies. And the key, which we'll talk about when I show you, when we just go over case study two a little bit, is really honing in on the question itself. Um, but just to update you on a couple of due dates, we have our case study four, which will open up after class. That'll be due June 30th, and then exam four, which will happen on July 2nd. And then that coming week between July 2nd and July 9th is when we should have some kind of communication with each other. So several of you have already set up meetings with me either by phone or Skype. So keep sending emails to do that, or let me know if you feel like everything's fine. It's nice if the cancer rough draft is turned in on July 2nd, then part of that mid-semester appointment can be discussing the cancer presentation to be sure that you're on the right track, because that is worth a lot of points. Yeah. I'm sorry, Dawn. I thought I sent everybody an email. So if you sent something to me and I didn't respond, re-email me because I might have missed it. The all case studies are always due the midnight before the midnight of the day. So if the case study, like this next case study, is due June 30th, then the case study is due June 30th by 23:59. So just be sure that we connect in some way, and it is good if we can look at that. Besides, talking about the class itself, if we can look at that cancer rough draft to be sure, because there's always a usual little bit of a growing pain in the beginning of this class as you kind of get used to what the expectations are, and this kind of class if you haven't been in it for a while. Yeah, I give feedback, just like, because you have to put it in the. So in terms of feedback for the mid-semester for the cancer rough draft, there is a Dropbox in Moodle for the cancer rough draft, and I will give you feedback there, as well as the points that you earn for doing the rough draft. But our discussion, we can have a specific discussion about it, especially if you have questions when we meet for that mid-semester appointment. What was I saying now? I forget. Oh, I purposely have backloaded the points in this class so that one of the two assignments with the largest point total are the cancer presentation and the final assessment, knowing that in the beginning things always are a little rough when you're trying to figure out exactly how to go about doing these assignments and getting back on track in a more biology, physical science basis instead of some of the other types of classes like research and policy and stuff that you'll take later. So there's definitely a sweet spot when you're answering these questions. You can give too little detail, that's for sure, but it seems to me that the problem is not too little detail. The problem is too much detail and not focusing in on the question itself. So I'm just going to share my screen because I want to look at the second case study a little bit. so that I can tell you what I mean. And in addition, offer you a chance to fix case study 3 before I read it. Sort of based on the feedback that you've already gotten for case study 2 and what we talk about right now. Like I haven't looked at case study 3 yet, so this would be the time to, try to alter it if you think that'll help you. So let me just um, tell you what I mean here. Okay, wait, where are you? Okay, so this was the case study, and I'll answer. Julie, we can talk about that separately. That's all inside the syllabus as part of the projects for the class. So, okay. Um, Here is that. Oh, I don't know if this is sharing. I don't think it is. Let's try that again.
Yeah, we can't see a screen yet.
Okay, that was my homework, so don't look at that. Okay, can you see now? It looks like I'm sharing now.
It's coming.
Okay, so here is that case study that I graded yesterday and the day before. So each of the scenarios show a patient some clinical information on a patient that has undergone some kind of tissue injury and has clinical manifestations of that tissue injury. So the first patient's injury is not an infection. There's been no skin breakdown. Or opening to the skin. This patient likely tore their muscle during activity. So they have, like a mechanical traumatic injury inside a compartment in their leg and have gone through both the vascular and cellular stages of inflammation. So the first question about this is, when you look at this case study, which symptoms are suggestive of the vascular phase? So this is key, and then explain those based on the underlying pathos. So this means that you are only going to focus on the symptoms that are related to the vascular phase of inflammation. And that is called vascular because obviously there's cells involved, but the issue is changes to the blood vessel that happen first, which make it the vascular phase. So those symptoms that you would focus on then, those symptoms that you would focus on then have to do with the vascular phase. So you should talk about heat, redness, and swelling, because and pain, those are the cardinal signs of the vascular phase of inflammation. And the patient has those, and the only thing that should be described is that. The issue where people had more of a problem, most people did fairly well on this question. It became more of a problem as this went forward. So again, we're looking at that same phase, and we're looking at symptoms that the patient has that are suggestive of the cellular phase. So that means do not talk about the vascular phase, because then the question to me is, can you separate the stages? Are you aware of which symptoms are really indicative of the vascular phase and which symptoms are indicative of the cellular phase? I want you to pull out information on one piece of the whole thing. So sometimes students have said, like, well, it's hard without giving some backstory. Well, if that helps you when you're making your notes, that's fine, but I don't want to see that. I don't want to read that. I want to read how you have interpreted this patient scenario in terms of the cellular phase of inflammation. And the symptom that's suggestive of the cellular phase is a large amount of exudate. And so even though during the cellular phase of inflammation, obviously you couldn't have it without the vascular phase, but that's not what we're talking about. So I didn't want anything from the vascular phase in there. And there are other cells that are pulled into the interstitial fluid besides the neutrophils. The neutrophils have the largest, they are the reason for the increase in exudate that takes place during the cellular phase of inflammation. So the whole answer to this question should only discuss how the neutrophils get there, which is the margination, transmigration, chemotaxis, phagocytosis. Then the neutrophils get there, and then the neutrophils' contribution to the exudate, which is that after they phagocytize material, they then necrose, spilling their cellular contents, and that thus increase the amount of exudate that's present. And so that question, there were many people that wrote way too much. It had nothing to do with the question itself. Yeah, that was a common thing. So then, then the question to me is, do you know what I'm asking you? And sometimes people are just, I don't know if you guys have a lot of trauma from your previous undergrad or college experiences, or that you're worried that you're not going to get points because the answer seems too short. Yeah, you all have PTSD. I don't know what it is, but I'm not trying to trick you. I want you to explain to me and hone in on the question itself. Yes, I think you guys all think I'm trying to trick you. It doesn't, I don't want to read 10 pages each case study. Seriously. No, I don't. So you need to focus on the question itself. And so lots of people had that issue. And sometimes people explained all kinds of things besides the cellular phase of inflammation. Then people lost a lot of points in this section, which is unfortunate because I think you knew what it was because you described it other places, just not where it was asked. The other issue was what kind of exudate it is. The patient bled. Right. No, I want a short answer. I seriously do not want to read 50 pages for each case study. I can't. The patient bled. So, because that's what happened. The trauma caused bleeding. And there is a large amount of ecchymosis. So there are wounds that will be very bloody. There's still the same things that happens. There's still the vascular phase. There's still leaking of plasma and proteins and blood potentially from the vessel. There's still neutrophils that come. There could be a huge hematoma there. Part of the job of the neutrophils is to liquefy that hematoma so that it can be, so it can get, we can get rid of that blood, that old blood. That's part of the job. So knowing that there is a large amount of ecchymosis to that area, the answer for the type of exudate is hemorrhagic. There were several people that just couldn't pick a side. You couldn't say serous and hemorrhagic because it's not both. It's got to be one. You can't play it both ways. You have to pick one. Yes, I mean, so it's that exactly what you're saying, Erica, is there is a sweet spot to this answer. You can miss something super key and then you're not going to get the points, but you can't just throw everything you know up against the wall and hope something sticks either. It's got to be serosanguinous. Not in this case with a huge bleed, Amanda. So that you gotta really hone in on the question. So yes, this is compartment syndrome because the injury occurred in a compartment that's wrapped in fascia that is not stretchable to accommodate the exudate. And luckily in the leg, the vessel is so large that the last thing that gets compromised for compartment syndrome in the leg is the vessel, but the nerve and the increased pain would definitely speak to compartment syndrome. So that was the answer for that. No trick questions. Okay, then the last one was where we really sort of derailed as a group. Um, because, so here we have this whole brand new patient that has gone through a different type of cellular injury. This is a cat bite. Cats are notorious for causing infections because they deposit bacteria deep into the tissue with their little peepers. And so this person has gone through, certainly has gone through the vascular and cellular stages of inflammation. But this question is specifically asking you, how do you know from that, what are the clinical manifestations that tell you the patient has gone into the third line of defense? How do you know that it's not just innate immunity now? We've crossed into adaptive response. And so if I look at the clinical manifestations, I have to pick out the manifestations that speak to adaptive immunity. And that does not include vascular inflammation or cellular inflammation. So sometimes people explain the whole thing over again. No, I don't want you to do that. The question is asking which clinical manifestations um, speak to adaptive immunity. Yes, sometimes the question will say, what are the positive symptoms? But sometimes it's asking you to clue into specific positive symptoms. So you really do have to read the question closely, because there are many positive symptoms in that last case study, but not all of them are related to the adaptive immune response. The only two that are directly related to the adaptive response is lymphadenopathy and high fever. The rest of the vital signs are indirectly related to fever. So the mentioning the rest of the vital signs was less of an issue to me. The issue was explaining all of the vascular immunity, explaining all of cellular inflammation, including the purulence. That's all. That's all. That has nothing to do with adaptive immunity. It's that there's antigen specific B cell differentiation and proliferation of B cell lymphocytes in the lymph node. And that amount of cellular division and increase in interleukin and cytokine release will increase the temperature far more than just macrophages would in the normal inflammatory response. So those are the two key parts to it. And then the point was, yes, but the question on that, the question itself doesn't say explain all the positive symptoms. The the syllabus was trying to let you know that the case studies were to be about linking positive clinical manifestations to pathology, but you have to read the question. And it seems like people weren't really doing that. They were just honing in on positive symptoms. So to get the full points for that question, I didn't take points off for people putting too much in. I will, however, start doing that though. But to get the points for that question, you had to you had to mention that there had to be antigen presentation causing differentiation of an antigen-specific B cell lymphocyte. So you couldn't mention, where you were not supposed to mention, each pathway to get to lymphocytes, because this isn't a viral infection. Well, the structure to follow is the question. The question is asking, I guess I'm confused about the disconnect myself, because the actual question says which symptoms correspond to adaptive immunity. So I don't understand what you thought, like, why would I then mark it wrong if you put other stuff? That's not what the question said. Additionally, if you have a question about the assignment, please email me. I prefer that than to reading a 10-page paper when that's not what I asked for. Oh my God, you guys, I was like, oh my God, I can't even take this. Um, so anyway, knowing that this was an issue and that there was some issues with interpretation of the assignment, or maybe not fully reading the question closely, or all of the above. Right, but it's still, I understand you're trying to explain to me what you were thinking, but I'm telling you to read the questions and answer the questions. Um, so please, I want you to go over the case study you turned in for this week. Please go over case study 3 and edit it, reading closely what the questions are asking you. The syllabus was just to let you know that the overall goal of the case studies is to link clinical manifestations to pathology. Each individual case study will have its own sort of flow, potentially. So now that we, right, so now that we know this, let us do that. So for case 3, because it hopefully would be, um, you know, something that should be easy to fix, if you could resubmit in the same Dropbox, because, oh, you're welcome, because, that Dropbox didn't close or anything. So go into case study 3, look at what you put in, switch it out, and you'll have till midnight on Sunday to do so, because I won't start looking at them until the weekend is over. And then you can edit them and be sure that the answer is what you want. And then we'll just go from there. But just so I did not take off any points for people that, I didn't take any points off if you added too much on this last case study. I only took points off if you didn't actually answer the question. So people still did lose points if they didn't actually answer the clinical manifestation that we were looking for. But otherwise, going forward, I will remove points for not answering the question that was asked, so that we are all on the same page. That was the same thing that happened on the exam that I noticed as well for exam 2, especially in the first questions, because the first question specifically asks what neutrophils do for wound healing. What do macrophages do for wound healing? And so some people, listed their job in terms of, phagocytosis, or, but they didn't actually put in the part that was specific to wound healing. So I think that even beyond the syllabus stuff, there is still like this concern about, thinking the answer is too short or something. I don't know what it is, but we'll go forward and just focus on what the question is asking. So you have, if you did not do, so these are the 2 announcements for you. If you did not explain what you predicted, if you did not explain the predicted sickle cell lab values for bilirubin and iron, please send me an email so that can be added to your exam. So then that kind of addresses, I can't remember which of you asked, like, what if, can I go in and add some? Only if it's necessary. So think about it before you would add to it. Because you really would only have to say if you thought it would go up and down and why. You don't necessarily have to, you'd have to just do a little background information, but you don't have to explain, that's a recessive gene, and then that means these parents got it, and blah blah. And you're like, you have to. I'm trying, Michelle. Kind of hone in on the down and dirty answer, especially on the exam, because there's several questions. You only have so much time to answer, and that can be the issue too. You're going to run out of time with the last question if you're spending so much time on the first one. So anyway, we go forward. So I will wait to grade case study 3 until after Sunday by midnight so that any people can do edits. So here's something funny, Amanda. I will have to say, people have complained about the multiple choice tests. I mean, there's no winning. I did short answer tests first, and people kind of liked them, kind of didn't like them. And then the thought was we really need to start doing more tests using ExamSoft, which is a protected test environment. And that we should get you guys ready because obviously the certification exam will be multiple choice. And so I did multiple choices. And then when we had to switch over to online all of a sudden last semester, people were having trouble getting on ExamSoft. So I switched it to these kinds of exams. I'm not sure what I'm going to do next semester, if I'm going to do like a combo or I will tell you that people weren't happy the other way either. That makes you feel better. Yeah, I mean that's kind of what the case studies are. People hated the multiple choice exams. They're hard. I mean, I think they're hard. If it was me, I would prefer the short answer because there's a little more wiggly room. People sometimes did not do very well on the multiple choice exams, and that was always an issue too. So, anywho, there's where we're at. Any questions about what's due? Oh yeah, the grades will go down if we did multiple choice exams. Completely true, Karis. They will. Especially the first exams. Oh my god, the tears. So, but we won't go there. We're gonna keep how we are because that's how we started. So, anyways, now we're gonna start talking about the nervous system, and the first thing we're gonna go over is just remembering the parts of the nervous system and how it's organized. Because, as always, many of the issues within the nervous system, whichever part, is really superimposed the inflammatory process and infectious process onto areas in the brain. So, you're welcome, Candy. So, we'll start with nervous system organization, and then we're going to review the stages of inflammation again, because we talk about it all the time. And there's special issues with the inflammatory process specific to nerves that increase the possibility of injury, and then we'll go over specific injuries. So that is the goal for today. So starting out, yeah, still sharing. We're gonna kind of talk a little bit about the organization of the central nervous system, peripheral nervous system, the entire nervous system, and the different parts involved. So usually people organize the nervous system between central and peripheral system. You can talk about it in terms of afferent and efferent nerves, and then somatic and autonomic systems are different ways that people organize this. There has always been an issue when talking about the nervous system, thinking about when you're actually not thinking about where the clear division is between the central nervous system and the peripheral nervous system. So we always have described the central nervous system as being nerves within the brain and spinal cord. The peripheral nerves leave from the spinal cord and go out to the periphery. So they do have a tiny amount of time that they're in the spinal cord. And so sometimes people confuse that. And this is something we're going to go over a bunch of times. But if that's someone's little brain and a nerve starts in the brain and crosses over and kind of ends at a level of the spinal cord, that's all the central nervous system. And then the nerve that's going to exit the spinal cord is the peripheral nervous system, But that peripheral nerve does spend a tiny amount of time within the spinal cord. But that's still the peripheral nerve because most of its time is in the periphery. Whereas this is the central nerve, obviously all of its time is in the central nervous system area. So I just don't want people to confuse that because that has happened and that's always very sad. Um, the afferent division is the sensory division. So a nerve in the periphery picks up all kinds of information, whether it be pain, temperature, position, and sends that information through to the level in the spinal cord that corresponds with it. And then that information is sent upwards. Efferent is after the brain interprets that information, interprets the environmental information, the brain sends a message down that's supposed to go out to the periphery to do something. So a message going into the central nervous system is sensory information that's afferent, and the message leaving is efferent, going to cause the body to do something based on the information that was given. This efferent side is further divided into autonomic nervous system, which is the sympathetic and parasympathetic nervous systems, or the somatic voluntary nervous system, which is the skeletal muscle. So this part of the nervous system we're going to talk about more when we talk about the cardiovascular stuff, when we talk about respiratory stuff, we talk about GI stuff, more when we're talking about when those systems are in use. This time, we're going to focus more on how information goes in and comes out and affects movement that we control. So, oops. The nervous system obviously has many jobs, basically controlling all of our movement and as well as integrating all of our sensory information, and should be where we have our substratum, or the area where intelligence, anticipation, and judgment occurs. The basic unit of the nervous system is a neuron, and that is how information is sent back and forth. And glial cells that protect the neuron and supply it with oxygen, take away carbon dioxide, help maintain electrolyte levels inside and outside. So those are the supportive cells of the nerves. The neuron itself has it can be described based on several parts of the neuron. So the cell body is where the cytoplasm is. This is where the dendrites come off, and the dendrites are what will pick up information from other nerves or environment, depending on which direction the nerve is going. The axon is branches off of that cell body and can be of variable lengths depending on, is this nerve coming from the spine all the way down to the feet? Or is this just an interspinal nerve that's only a couple millimeters? So that axon can vary. And at the very end of the axon is a synaptic terminal. And inside that is where all the neurotransmitters are stored. And then that information neurotransmitter can be released. These are different shapes of neurons depending on where they're located. Glial cells, or those supportive cells of neurons, are named differently and kind of vary in complexity depending on if the nerve is within the central nervous system or is primarily in the peripheral nervous system. The central nervous system has several different types of glial cells. Astrocytes, which fill in the space around neurons and blood vessels. Oligodendrites, which are what create the myelin for the nerve, and myelin is sort of that insulation that can be work its way down the axon. Microglia, which are kind of the sort of monocyte type cell, of this area that can exercise phagocytosis and sort of clean up cellular debris. Ependymal cells are special cells that line the cerebral spinal fluid cavities. So those are specific type of cells in that area. Whereas in the peripheral nervous system, there's only two types of glial cells. The Schwann cells, which are similar to oligodendrites in that they produce and support myelin, and satellite cells, which are kind of like the astrocytes in that they help maintain electrolyte balance and help with carbon dioxide and oxygen exchange and stuff. So this is a picture of them, and certainly you can see the change and complexity between the two. This side is obviously the peripheral nerve that has some satellite cells that are protecting the cell body, and then the Schwann cells, which will help maintain the myelin in the peripheral space. But here in the central nervous system, these are ependymal cells are lining the cerebral spinal fluid. Obviously, the oligodendrites are more complex than the Schwann cells, astrocytes, and microglial cells. It's a lot more going on in the central nervous system. So down the axon is where the nerve signal or the action potential will be propagated. So this means we have to remember the first class where we talked about the action potential. And when we talked about it just in a cellular level, the major intracellular cation was potassium, and then there's a small amount of potassium outside the cell. The major extracellular cation was sodium, and then there's a smaller amount of intracellular sodium. Normally, the cell membrane is negative. When there is stimulus to the cell, the sodium channels open, which allows rapid influx of sodium. Which increases the positive electricity in the cell, which is generation of the action potential. And then as the cell returns to its resting state, the first thing that happens is potassium starts to leave the cell down its concentration gradient. And then we quickly get back to resting potential by the activity of the sodium and potassium ATPase pump, where we pump sodium and potassium against their electrical potentials. And we reestablish resting potential until the next signal comes and sodium channels open again, and we do the whole thing over. So if this occurs down an axon, it'll occur in a stepwise fashion. We could sort of divide the axon into segments, and that action potential or the stimulus could come down to the cell body and cause sodium to come inside a segment of the neuron, which will quickly increase that's positivity, that is the action potential, and then eventually potassium will come out, and resting potential will be reestablished at the same time that the signal goes down further, kind of marches down the segment of the axon. Sodium comes in, the action potential is reached, we start to go back to resting potential, and then this segment depolarizes, and so we go followed by repolarization. We kind of go and march down the line. If we wanted to go faster, because this will go slow, we would slowly depolarize and then repolarize each segment of the axon till we got to the end. The reason to make the myelin is sort of like this, down the length of the axon. This way we can jump segments. So here's where we had sodium depolarize, and then eventually we'll repolarize. But instead of having to go down a specific segment and march our way slowly down, we can actually jump over space, and so we can change the action potential. We can increase the speed of the action potential down the length of the axon by going in between the myelin spaces in these what are called nodes of Rainier. So they're allowing us to jump space so we can go faster. So that's the point of myelin, as well as being an insulator and a protecting, kind of shield for the axon itself. So the whole point of generating the action potential and having it propagate or travel down the axon is to get to the synapse, to the presynaptic terminal. So if this is the end of the axon, and then here's the synapse, this is the presynaptic terminal, this is the synapse, that's the empty space before the next thing, either another nerve cell or a muscle or a gland that's part of the autonomic nervous system. Depends on what the next thing is. If it's a nerve, then this is the post synapse, and this is, we said, was the presynapse. If the action potential is allowed to go down the entire length of the axon to where the neurotransmitter is stored, the release, this action potential getting down here will allow release of the neurotransmitter into the synaptic space. So here it is. That neurotransmitter could be excitatory, well, something like that. Bad spelling. Excitatory, meaning it'll send that message to keep going, keep the action potential going to the next thing. Or it could be inhibitory. It could say, turn everything off. But that neurotransmitter will interact with receptors on the next thing and either continue the action potential down, or it could stop it from going forward, depending on what kind of neurotransmitter it is. So the presynaptic terminal is where neurotransmitter is synthesized and stored. The postsynaptic membrane is the neurotransmitter's receptor. And then eventually we have to get rid of the neurotransmitter. So here we go. We let the neurotransmitter out. It went to the next thing and interacted with receptors. And now whatever we wanted to happen happened. We want to clear this synapse. That's the next part. There are 3 ways to clear the synapse. We can do something called reuptake, which might sound familiar because that's the basis of a lot of drugs that are advertised on TV. We can increase recycling of the neurotransmitter that was in the synapse, so we can get rid of it that way. Some of it will diffuse out into the surrounding space between nerves potentially and be, and astrocytes or other cells will get rid of it. And some of the neurotransmitter will be inactivated by enzymes that live there. And so obviously this is important because these are areas where there could be potential pathology, which obviously we'll talk about coming up. So we're going to stop here and take a break. And we're going to start working our way down the nervous system, just kind of going over the parts of the nervous system so everyone's clear on what every part does. So it's 5 to 8 right now, so we'll go to 8:05 for a break, and then we'll come back.